Welcome to section 13.1, where we're going to start focusing on the idea of how we control organisms to get traits that we want to become part of that organism. So when we're talking about agriculture, when we're talking about pets, when we're talking to some extent about medicine, we are going to have instances where we want certain organisms to get traits that they may not naturally have, or maybe a certain variety of them does not naturally have. So maybe there's some peas that are disease resistant, but not others. And so we want this particular type of pea, maybe, I don't know, a sweet pea, to have the disease resistant that normally a different type of pea has. And so we've got to come up with some way of making that happen. Now, the most simple way, the oldest way, is something called selective breeding. Now, this makes a lot of sense. If an organism has a trait that you desire, you breed the individuals that have the best version of that trait with one another. And so that way the next generation tends to have individuals that have more of that trait and more pronounced versions of that trait. And you can continue doing this over and over until you get what you desire. So some extreme cases are if you want a small dog for companionship, by breeding only the smallest dogs. The other dogs you essentially just get rid of, we'll assume adopting out, nothing cruel. And then you just keep then taking of the offspring of those smallest dogs, just the smallest of those. And so over time, over many successive generations of doing this pruning, we can now get very small companion dogs like a Chihuahua or a Pomeranian or a Pug. We can also do the same thing for agricultural benefits, where we can try to get cows that are either more meaty or produce more dairy, or we can go through and try to get chickens that lay more eggs or put on weight faster. So this can be applied for a whole bunch of purposes. The upshot of this is kind of the old adage where you'd have what they used to call seed corn or seed crop, where you take the best organisms. Let's say we plant corn, you'd take the best corn plants and you'd save their seeds and you'd eat the other corn plants, the ones that weren't so impressive. And so that way, by planting the seeds, the corn from the very best plants, you'd help ensure that next year's harvest would be better than this year's, because the plants are likely to do better because they came from your top producing plants. So the next generation should also be higher producing plants. So you just do this over time, kind of in a logical fashion, and we eventually get some organisms that are rather amazing, whether it be crops, corn that produces tons of kernels, or whether it is something like cows that ultimately can produce gallons of milk in some cases per day, which would not be normal in the wild where they'd conserve energy and not do that. But we've helped them out. Now, if we're trying to introduce a trait that did not exist in a particular type of organism, one way to do that is hybridization. If we can find another similar organism, one that's either of the same species, it's just a different breed or strain of plant or animal, or in some cases you can have a very closely related species where if we put them in non-natural conditions, like a lab, we can entice them to reproduce so we can get a trait from that other organism into our desired organism. So this could be something as simple as taking a particular fur color and we want to get a dog type, I don't know, a Chihuahua that has, orange fur like a golden retriever. You could then try and take some Chihuahuas and figure out a way, maybe artificial insemination on this one, and ultimately try to get one where it reproduces with something like a golden retriever or some other dog that's maybe a bit smaller that has that particular fur color. And so by doing this, we can try to bring in that new allele, that new gene, that new version of something into this existing organism, the Chihuahua, so we could eventually get this red Chihuahua. Now you can also take in we use this a lot in agriculture for things like disease resistance. We can use it for particular things like in sheep or something. You might want to work on the wool because you're using it for wool production. We're normally going to be doing this for some type of specific benefit. You'll see a lot of this isn't so much for the pet industry. It's more going to be for our agricultural purposes like you see with corn here. If we can find something that ultimately grows faster, if we can find something that's more drought resistant, if we can find a version of a plant that is more disease or pest tolerant, then at that point we want to get those genes, we want to get that ability to cope with all these conditions that normally might kill the plant. We want to get those genes into the plant, and so that way it allows us to benefit because the plant's going to live better, the plant's going to ultimately reproduce better, the plant's going to give us more food. Same thing would go for animals. If we can get them to grow faster using less food, that's a great boon, so that we have to feed the cattle less, but we get more meat. Any of those things are great. Now the problem is, because you have to do this over generation after generation, you've got to keep track, you've got to do records, you've got to make sure you're buying or getting the perfect animals that'll suit your needs. You know, they've got to have the right traits that you can track. This is ultimately going to be fairly expensive to house everything and keep track of all this, and it's time consuming because this can take generations and generations to get exactly what you want. And so this is not always going to be ideal if you want something in the really near future, because this process, especially if it's occurring in a long-lived species where it takes a while to get the animals to maturity, to like know what their actual physical condition is going to be, this could take years and years and years, maybe even decades, to actually come to fruition and benefit us. So that's the major drawback with this. But it is still relatively low tech. It just takes time and good facilities. Now, when we're trying to make specific organisms, when we're trying to do this process of, getting this particular ideal version of an organism, we're normally going to have to do a fair amount of inbreeding. So I know we've typically talked about inbreeding being generally bad, because when you do inbreeding, it tends to make organisms more likely to be homozygous. And because there's a lot of recessive traits that are out there that are bad, if you become homozygous for them, you can be affected by those recessive traits, and that can be very bad for the individual. But ultimately, if you're trying to make a new breed or a new strain, you typically have to do this because there's only so many animals that you have that meet the particular criteria you're trying to breed for. So when you get several of them, you have to interbreed them because you want the offspring to look like them. So inbreeding is going to make organisms favor being homozygous. Now, once again, this can be bad if you're going to get some of these recessives we talked about that can be negative or lethal recessives. If they become more common, this can be bad. But you can also make the organism be homozygous for the good traits. And if you're homozygous for the good traits, you can't pass on those bad traits. So doing something like inbreeding is not inherently evil or bad. It doesn't need to be. But you have to be careful when you're doing this. You have to keep track. That's why when we do inbreeding, you see people focus on pedigrees so much. And those pedigrees matter because they track whether or not these individuals have any type of propensity for a bunch of these negative traits. Because you're trying to ensure that the animals that you breed as a reputable breeder are homozygous for all the good traits, that you're weeding out all those heterozygotes that might carry the negative traits. That's why if you go to a breeder, so many dogs get sold, if we're talking about dogs for instance, as pet quality because they can see that they either have an issue or they're worried that they might be a heterozygote and they might be able to pass on that issue. And so they therefore sell them as non-breeding to make sure that they don't kind of deteriorate the genome of that particular breed. And so by only focusing on the best individuals, the ones that are homozygous for all the good traits, we can make sure that inbreeding is not going to be negative in this case, other than the lack of diversity it creates. You can argue that's always going to ultimately be somewhat of a negative. If you end up being homozygous, even if it's for the good traits, that's the good traits for right now. That doesn't mean if things change that that will be a good trait later. If you wonder why there's a picture here, this is Charles II. Ultimately, his dad married his niece. So he technically, his mom was also his cousin. You can tell by the way he looks, probably not the best guy. He did have some physical and mental issues, which were likely the result of his inbreeding. So once again, if you're going to do inbreeding, you do have to make sure you're careful about it, or else bad things can happen, like Charles II. And then lastly, if we're going to be careful about who we're breeding, you need to understand the basic idea of a test cross. Now what a test cross is if we have an individual, and let's say they're dominant for some particular trait. So we know they have at least one big Y, we'll say. We'll assume it's pea pod color, doesn't matter. But if you know that they have the dominant trait, you know they at least have one big Y in this case. But we don't know what the other one is. And so we need to figure this out, because if they're a heterozygote, we don't want to breed them. If they're a homozygous dominant, then we do want to breed them. So one way of doing this is called the test cross, where you breed the individual that we're unsure about, so it could be kind of this version A or it could be version B, and we breed them with a homozygous recessive individual. By doing this, if we see any of the offspring are recessive, we know that the unknown guy must be a heterozygote. If we see absolutely none of the offspring are recessive, then there's a very good chance that the individual that's unknown is homozygous dominant. And so this is why you might have an individual you're not sure about, and so you do one early breeding with an individual that's recessive, and if it's something that produces lots of offspring, like many plants or many animals that have like a litter, You can then look at those offspring and try to see if there's even a single recessive. If there is, you know it's a heterozygote, and so if it's something you're trying to weed out, you would not further reproduce that organism. If that organism produces no recessive individuals, then there's a very good chance that it's homozygous dominant, so that individual might move into your breeding program and you would continue to breed it as long as you could, essentially, so you could maintain those quality dominant genes because it's homozygous for it within that gene pool.